Okay, hey guys, this is Annalicia.
And Darshawn.
And we have a video for y'all on genetics for biology. So let's jump right into it. So first we need to know that y'all know some key terms because these key terms are going to be in the video talking about different types of topics throughout Mendelian genetics. So first, a gene. This is pretty much the basic thing that you're going to need for genetics. It's in the word genetics. A gene is a segment of DNA that is responsible for the inherited trait of an individual. And so there are two types of genes. You have dominant and recessive. The dominant is always fully expressed whenever the trait is present. Whenever you see it, it's going to be expressed. And a recessive gene is only expressed when the dominant trait is not present. So it can only be by itself. And then you have an allele. And this is a different variation of a particular gene. This is where you have homozygous dominant, heterozygous, and homozygous recessive. And then you have genotypes. This is a combination of alleles. And so two different genotypes you have are homozygous and heterozygous, like I just said for an allele. And then a phenotype is the expression of an individual's phenotype as a physical trait in the organism. So phenotype, remember, pheno, physical, genotype, gene. And so you'll need these for the rest of the video. So to begin with Punnett squares, we're just going to start with a basic monohybrid Punnett square, which is essentially the foundation for all Punnett squares when you go into dihybrid and trihybrid Punnett squares. So we're going to start with dominant trait, which can be said, we're going to do dominant homozygous. So we're going to do BB. These are both capital because that's what dominants are. And we're going to do recessive, which will be, we're going to say BB, lowercase b and b. So how we're going to put that on our Punnett square is, on one side you have one organism's alleles, and then on the other side, or at the top, shall I say, you have the other organism's alleles. So both parents' alleles go into the Punnett square, and then you're just going to cross them. So in each one, you put each box, and then you go and add the recessive alleles. Now, analyzing all of these boxes, each one is going to be tall. Why? Because that dominant trait is in each box. Dominant is essentially that all dominant alleles will show. Because of this, every organism can be defined as tall versus short. And that's how monohybrid Punnett squares work. So, delving a little bit more deeper into Punnett squares, we go into dihybrid crosses, which follow monohybrid crosses. So this is a little bit more complex, which is why you have an allele key. So, your dominant trait is tall plants. This can be signified by a capital T because your dominant alleles are always capital. And then its recessive trait are dwarf plants, and this is signified by a lowercase t, which is always the signification for a recessive trait. Then the same can be seen in the dominant trait. This is the second trait, which is purple flowers versus white flowers. And the purple flowers are dominant, so they're seen by capital F, and the white flowers are recessive, so they can be seen by a lowercase f. Now, then you go into genotypes. Your genotypes for your first parent is tall and purple. This can be seen because it's a homozygous dominant trait for both of them, so it will be tall and purple. The same is, well, not so the same for parent 2. You both have recessive traits for parent 2. So this is seen by lowercase t, lowercase f. So it's short and white. So FOIL for your parents is similar to kind of distributive property in algebra. Science and math go hand in hand, which is why I keep referring to algebra. But basically what you do is you distribute this T to each allele, and then you do the same for the second one. So once you do that, you get T, F, T, F, T, F, essentially, well, not essentially, the same for all of them. This is because when you link this, they basically combine. You get new combinations. The same goes for FOIL with your parent 2. So you FOIL again, that goes there. This T goes to that F, and this T goes to that F. You essentially get TF for all of them. And so that's essentially how you get your new combinations so that you can put this in your dihybrid cross. Okay, so our question, I forgot to add it to the last slide, was cross a homozygous dominant parent flower with a homozygous recessive parent flower. I mean, that was pretty much common sense, but I still wanted to make it known. So, getting a little bit more complex into crossing dihybridly, you will essentially cross how you do with a monohybrid, but you make sure that your alleles line up with each other. So you wouldn't do lowercase t and capital F. No, that is totally not right. You would line it up with its respective allele. So you would do dominant T with recessive T, and then dominant F with recessive F. You just keep doing that across the entire table. And so on. So after filling in the entire square, we see that we have the literal same allele combination for every single box. And so we have to fill in our genotypic percentages and our phenotypic percentages. Remember that I told you genotypic percentages are the percentages that you look at internally. So how your genes are wired inside of you. That's genotypic, how your genes look. Whereas your phenotypic percentages are how you look. So, for your genotypic percentage, you see the same thing across the entire square. So you see T, T, F, F for every single square, like everything. So for that would be 100% because everything is 100%. Phenotypic, if it's the same thing, then it's going to be the same thing for the phenotypic. If dominant, you see a dominant allele in any square, it's going to be that trait. So dominant for the dominant for T was tall, and dominant for F was purple. So they're all going to be tall and purple. So that is 100% tall and purple. And that is how you fill in a dihybrid Punnett square. So that sums up Punnett squares.
Hey guys, this is Darshan, and I'm filling in for Anna for the rest of the video. We're going to be talking about incomplete dominance, codominance, and sex-linked trait. So let's get right into it. Incomplete dominance refers to a genetic situation in which one allele does not completely dominate another allele, which therefore results into a new phenotype. For example, when we have a homozygous dominant blue flower with two big B's, and a homozygous dominant yellow flower with two big Y's. This can result into a new phenotype of green, since blue and green mix together, and this would be a heterozygous big B, one big B and one Y. So it'll have one big B from blue and one big Y from yellow. So when we put this into a Punnett square and we cross them over, big B will go down here and Y will go right here, and this will be repeated throughout the whole Punnett square. So big B, big Y, big B, big Y, big B, big Y. So the genotype of this new plant will be 100% big B, Y. And the phenotype will be 100% green, since blue and yellow together make green. So now we're going to go over codominance. Codominance is almost similar to incomplete dominance, but instead of having a new color, you would have patterns. So this is why some dogs or like panda bears are spotted instead of being one complete color. It is a form of dominance in which alleles of a gene pair and a heterozygous are fully expressed, thereby resulting in an offspring with a phenotype that is neither dominant nor recessive. So in this example, we have a homozygous dominant white bear and homozygous dominant black bear. So when you combine these two together, you would have one big B from the black bear and one big W from the white bear. Instead of creating gray, you would have different spots. So if you look at this example right here, you would have some white and then some black on this panda bear. Finally, we're going to go over sex-linked traits. Sex-linked traits are a trait associated with a gene that is carried only by the male or female parent. So a female is expressed by 2 X chromosomes. So this is your mom. And then a dad is expressed by 1 X and then 1 Y chromosome. So how sex-linked traits work are, it has like almost like an exponent on their chromosome. So if you have, say, because we're going to use hemophilia, so we're going to use an H as our chromosome, as our exponent. So big H means you don't have hemophilia. Little h means you do have hemophilia. But if you combine them together, so if you have 1 X with a big H and 1 X with a little h, you don't have hemophilia, but you carry hemophilia in your trait. So it won't affect you physically. So this would be, so if you have 2 big H's, you're called normal. Oops, spelled that wrong. Oh well. If you have 2 little h's, you have hemophilia. But if you have this, then you're a carrier. So you can carry hemophilia onto your next offspring. So if we combine a hemophilia mom, so she has 1 X with a little h and another X with a little h, and a normal dad, so he has an X with a big H and a Y. This new offspring will be a carrier because this H is dominant. So this will be H, X, H, because this comes down and this goes this way. This will be X, H, Y, X, H, Y. So this will be 50%. This part right here, these 2 X's, there will be female. This will be a male. So the female will be a carrier of the trait. So 50%. 50% is female. And then 50% will be male. On the male side, the offspring will have hemophilia because there's only a little h. So 50% will be a carrier. And 50% will be hemophilic. And also females are the only ones who can be carriers because they have 2 X chromosomes. So a male either has to have hemophilia or they can't have hemophilia.
Okay guys, so that's all we have for Mendelian Genetics today. I hope you guys enjoyed this video, and I hope if you didn't completely understand it or enjoy it, I hope you find understanding from another source in your biology class, because biology is one of my favorite subjects, genetics being one of my favorite topics. So this video was pretty fun to make for me and Darshan. So in the end, pretty much, thank you guys for watching and enjoy biology. Bye.